I am Dr. Madhusri Haldar from Department of Botany, Sri Rampur Girls College. Today we discuss about Palaeobotany and Palynology. Our topic of discussion is Pollen Morphology and Pollen Aperture. Before going to detail, we have to know what is Palynology. Palynology means that is a branch of science dealing with the study of both extinct and extant pollens and spores. Actually, the term palynology means study of dust, and the term was first coined by the scientists Hyde and Williams in the year 1945. There is a diagrammatic view of a young pollen grain and a mature pollen grain. Here, the difference is very distinct. In the mature pollen grain, the wall of pollen grain is very much thick and it is ornamented. But in case of young pollen grain, the wall is very simpler and thinner. So, I use two different terms, that is spore and pollen. What is the difference between the pollen grain and the spores? First thing is that spore is the unit of asexual reproduction in lower group of plants such as algae, bryophyte, fungi and pteridophyte. That means the embryophytic plant. But pollen grains are male or microgametophytes or seed plant which produce the male gametes or the sperm cells. Second point of difference is that according to the size of spores, there are two types of spores. That is, one is microspore and another one is megaspores. Microspores are comparatively smaller than that of megaspores, and from microspore male gametophyte are produced, while from megaspore female gametophyte are produced. In case of pollens, that always give rise to male gametophytes only. The third point of difference is spore can be sexual or asexual, but pollen take part only in sexual reproduction. And the fourth difference is that, and very much important difference, spore germinate through its distal pole and pollen grains germinate through its proximal pole. Now start the main topic, the morphology of pollen grain. Generally, the characters of pollen and spores are studied under following categories. Number one is polarity, that are of two types, proximal and distal. In the next slide, we discussed about the polarity in detail. The second character of pollen grain is the symmetry, that is of two types, radial and bilateral symmetry. And the third important character of a pollen grain or spore is aperture. Basically, the aperture is of two types, simple and compound aperture. Simple aperture is again of two types. One is porate and another one is colpate. Compound aperture also divided into two types, that are porate and colporate. So before going to the polarity, symmetry and aperture, that is the character of pollen grains, we have to know about the development of pollen grain from a pollen mother cell. Here PMC is equal to pollen mother cell. We know that pollen mother cell is diploid, or the chromosome number is 2n. By reductional division or meiosis, it produces 4 pollen grain or pollen tetrad of n number of chromosomes. So their arrangement are different types. Here we discussed about two different arrangement of tetrads. One is the isobilateral tetrad and the another one is the tetrahedral tetrad. In case of isobilateral tetrad, the pollen grains or tetrads are arranged like a orange. The orange cells arranged just like that the tetrads are arranged in that way. But in case of tetrahedral tetrad, 3 pollen grains are in one face or one plane and another one on the top of these 3 pollen grain. More prominently, I'll show you, that is the arrangement of isobilateral tetrad, that is side by side like the orange cells, and when we separate out these 4 or the tetrads, we will get this type of unit, and this type of aperture or mark will produced on it. That is, the monolith mark was produced in case of isobilateral tetrad, but in case of tetrahedral tetrad, that is the arrangement type, 3 pollen grains in the bottom and another one is on the top. Here we can get this type of units when we separate the tetrad, and this type of lead mark or 3 lead mark was produced on the pollen grain, that is known as tri-lead mark. Next come to the polarity. If we consider the isobilateral tetrad, then we will get this view. Actually the side view is that, and that is the polar axis, and that is the equatorial axis of the pollen grain, and this is the equatorial view actually, and these are the different pole. One is proximal pole and another one is distal pole. So from equatorial view we can see both the proximal and distal pole. The proximal pole is actually the pole nearer to the aperture, and the distal pole is the distal the position of distal pole is in the distant part from the aperture, and that is the polar view of isobilateral tetrad. And from polar view we can see only one pole, either proximal or distal, and here that is the monolith mark. In polar view actually we can see one pole proximal, that is a proximal pole, and only the aperture. But in case of the tetrahedral tetrad, that is the pollen grain, and that is the polar axis, and this is the equatorial axis. Distal pole is that, and that is the proximal pole. This is the equatorial view of a tetrahedral tetrad, and that is the polar view. Here we can see the tri-lead mark and the proximal pole only in polar view. So depending upon the tetrad type, different lead mark or aperture is formed on the pollen grain. Next is the symmetry. In case of the radial symmetry, the pollen grain is actually circular type, and these are the different poles of the pollen grain, and this is the bilateral symmetry of pollen grain, and in the left side that is a radial symmetry. Actually the difference between that is in case of radial symmetry we can get equal halves when we cut it down or divide it in any plane. But in case of bilateral symmetry, we can get only one plane from which it when cutted we get the equal halves. So that is about the symmetry of pollen grain. Next thing is aperture. That is very much important for our study. We already know that there are two types of aperture. One is simple aperture. So suppose that is a pollen grain and that is a simple aperture on the pollen grain, and if we zoom it out, the pollen wall is like that, that is bilayer pollen grain. One is intine and the outer one is called the exine. Then the aperture is obviously on the two layer, on the intine or the inner layer and also on the outer layer or exine. So the ecto aperture is that, and that is the endo aperture, or it is called as aura. So that is a simple pore on a pollen grain. If the length and breadth, actually length is to breadth, if less than equal to 2, then it is a pore, and when the length is to breadth of the aperture is greater than is equal to 2, then it is a colpa. That is, the length is greater than the breadth and the aperture like boat shaped. Then compound type of aperture. Here that is the pollen grain and this is the colpate type of aperture on the outer layer, and there is a aura or pore type of aperture in the inner layer. So ecto aperture is a colpa and endo aperture is a aura. So totally that is aura in colpa or colpi. That's why it is named as colporate. While on the other hand, another type of compound aperture is ecto aperture is a pore and endo aperture is a aura. That means ecto aperture is a pore and endo aperture is aura. That is, aura in pore is equal to pororate. Now come to the portion, that is the types of colporate aperture. That is pollen grain, and this is colpate type of aperture. That is actually the ecto aperture, and the endo aperture is a aura, but that is elongated to parallel to their colpa. And on the other hand, the ecto aperture is a colpa, but the endo aperture is a aura that elongate right angle to the colpa. So the thing is that one is parallel to the colpa, another one is right angle to the colpa. So the name given to it, low longate and la longate. Here low means parallel and la means at right angle. So there are two different types of colporate aperture. One is called low longate, another one is called la longate. There are many more types of aperture actually, but for the general student or past student, this type of apertures are sufficient. I repeat, there are many more types of aperture, but that is sufficient for the general student. Now come to the portion of NPC classification of pollen grain by Ardman and Straka in the year 1961. Here N represent the number of aperture, P represent the position of aperture, and C represent the character of the aperture. So basically NPC represent number, position and character of the apertures on pollen grain, and this classification system is actually have very much importance to taxonomy, or nowadays it is very important to the taxonomist, because depending upon this classification the phylogeny, identification and many things are done by the taxonomy. So that is the overall diagrammatic view of NPC classification. The first row represents the number of aperture on the pollen grain, and that is represented by the alphabet N. Second row is the position of aperture represented by P, and the third row is character of aperture and is represented by C. In the first row, N0, that named as atrium, that is the number of aperture is not clear, that's why it is called atrium. In N1, the number of aperture is single, that is monotrim, and in N2 the number of aperture is 2, that's why it is known as ditrim. In N3, that is number of aperture is 3, it is known as tritrim. N4 is tetratrim, N5 is pentatrim, N6 is hexatrim, N7 is polytrim, and N8 is anemotrim. In case of N8, the apertures actually present in a circular or reticulate type. Next is the position of the aperture, that is the first column is P0. That means the position of aperture is ambiguous, that's why it is represented by P0. P1 is called catatrim. In this pollen grain, the aperture is present on the proximal pole of the pollen grain, while P2 represent the apertures, one at the proximal pole, another one is on the distal pole, that's why it is known as anacatatrim. P3 represent the position of aperture on the distal pole, it is named as anatrim, and P4 represent the position of aperture on the equatorial zone, that's why it is named as zonotrim, while P5 represent the position of the aperture on equatorial view but in two line, that's why it is named as dizonotrim, and pantotrim represent the position of the aperture throughout the pollen grain, on the surface of the pollen grain. Next and the last row represent the character of aperture. The first is C0, that means the character of aperture is not clear to the scientist, and next is C1, that is the character of aperture is actually not true aperture, here aperture like just impression. Next is C2, that is trichotomocolpate, and that represented by C2, that means the number of aperture is 3 and the type of aperture is colpate, and these three colpate actually are joined with each other, and C3 represent the colpate type of aperture, C4 represent the porate type of aperture, C5 represent colporate, that means aura within colpa, and C6 represent the aura within the pore. So that is the character of the pollen grain from C0, that is ambiguous, and C6 is porate. So that is the overall NPC system of classification. I think that is very much helpful for the student, and that's all for today. Thank you.